It's just there to, clog up the receptors. This happens a lot when you get when you talk about muscles, when you talk about um, venoms or poisons. Some of these venoms can block these receptors in your muscles, and that can paralyze your muscles because they cannot be activated anymore. Also, this gets you can have different types of receptors. We talked about this. How cells can respond because they have the receptor or not have a receptor. But the same is true if you have different types of receptors. So this is how you can have one molecule. So here we have epinephrine. Epinephrine is also known as adrenaline. So when you're in the fight or flight response, you have adrenaline kick in, you have epinephrine kick in, it's surging through your body, and physiologists have found out, and we'll talk about this more next week, that epinephrine has different effects on different parts of your body. So for example, epinephrine can constrict blood vessels. They constrict blood vessels in your intestines, but they can also dilate vessels, say, in your muscles. This helps redirect blood flow to your muscles and away from your stomach or your GI tract. And so it puzzled scientists for a very long time. How does one molecule get you opposite effects? And the way you do that, we found out eventually, is we have different receptors that can still bind the same signaling molecule, but because they're different receptors, they lead to different effects in the cell. Yeah. So let's talk about signal and control. We'll talk about this more tomorrow, but these are a couple of concepts that are going to be talked about more in the future, so I'm going to bring it up now. So there's two types of control we talk about mostly. We talk about tonic control. And tonic control basically is the control of how often you're going to signal a molecule or signal a tissue. So in this example, this neuron is giving its signaling molecule at a rate like this. So every tick here represents a release of this signal, and it's affecting this blood vessel. So as time goes on, it's giving a steady beat of signal, kind of like a drum beat. But tonic control is about about varying this frequency. So tonic control is if you want to have an increased effect on the tissue, you can increase how much you are releasing signal. And so by doing that, you are overall exposing the tissue to more of that signaling molecule. And so the idea here is with every release from that neuron, you give a set amount of molecule, and the only thing you're changing is the rate you're giving it out. If you give it out at a really high rate, you're going to put a lot of molecules out there. And that can create effects. So this is, for instance, vasoconstriction. But you can have the opposite. You can slow down the rate of how often you expose a tissue to the signal, and that can have a different effect. And so a lot, especially for vasoconstriction, the amount of control you have will vary. So control, let's say this. The response is going to be proportional to the frequency of the signal here. Does that make sense? No? Yes? Maybe so?
Can you explain that one more time?
Yeah.
Like, why is it that the more signal is constricted, but then the less signal it's...
So in this example, we can say the diameter of this vessel is related to the concentration of the signal. You can just say that. Low signal is going to be related to a large diameter, and then and a high concentration of the signal can lead to a small diameter. And so for a neuron, every time it releases the signal, it's only setting out a set amount of signal. It doesn't control how much of the molecule it puts out for every, for every event. For every event, it just gives out X amount. So the way that they can control the amount, the concentration of signal that's being exposed to the tissue is by increasing the rate or decreasing the rate of how often they give the signal out. That's the idea of tonic control. By changing the frequency, we can change the concentration of that signaling molecule. Question?
So it just relates in terms of like muscle.
So this can be muscle. Muscle can do this too. In this example, this is blood vessel. You can see this a lot. It's not doesn't happen in every single type of tissue, but it happens a lot in a lot of tissues. One example is muscle. You can have multiple stimulations in the muscle, and another example here is blood vessel. Does this make more sense? All right.
So in terms of if contracting is fluidistic, is it like affecting the sarcomeres?
So muscles are a little different. So muscles and we'll talk about that in a couple of weeks. Muscles will contract when it gets a stimulus from a neuron. Now, that contraction can get greater in strength if it gets a higher frequency of stimulation. So it's a little different, but it is under the same kind of tonic control. You can control the tension of your muscle depending on the frequency of the stimulation. You can have antagonistic control. In this situation, you have two opposing signals, and so this is exemplified by a sympathetic neuron and a parasympathetic neuron. Sympathetic neurons are used for, the fight or flight response. And parasympathetic nervous system or neurons are used for basically R&R, rest and relaxation. They're making sure your body is in a conservative state. It's conserving energy, basically just chilling. And so these often are antagonistic because they want different things. A sympathetic neuron wants to increase heart rate, while a parasympathetic neuron wants to decrease heart rate. So oftentimes, the heart rate, if this is heart rate over time, it will increase or decrease depending on what is the more dominant stimulus. So at this point, we can say the sympathetic neuron was more dominant. It was exerting more control over the heart than the parasympathetic neuron. But in situations where the heart rate dipped, the parasympathetic neuron was dominant. The idea is only one. It's a game of tug of war, and what side is winning will dictate how the tissue is going to respond. The sympathetic neuron is winning the tug of war of control here. It's going to increase the heart rate. If the parasympathetic neuron is winning this game of tug of war of control, it's going to decrease the heart rate in this example. And if they're stalemated, the heart rate's going to be somewhere in the middle. And so that's the idea of antagonistic control. We see the same thing in insulin and glucagon. If you remember the fasted and fed state, those are mutually exclusive states, and when one was present, the other was not. That is another example of antagonistic control, where one thing is dominating, represses the other side, and vice versa. The idea is there's just two different signals, and basically fighting over which one takes over the response.
One is being blocked.
Sometimes it's being blocked, but sometimes it just overrides the other one. In this situation, it's all about how strong, how intense is one signal versus the other. This is basically what I drew. So yeah, if this one wins out, heart rate slows down. If this one wins out, heart rate increases. Okay, let's get to signaling pathways. I'm almost done here. I'm probably going to continue this next week, next lecture, but let me at least introduce it a bit. So we talked about, there are molecules that are sent out in the body to signal cells to do something. There's a whole cell transduction to how to get the signal from the outside to create a change in the inside. But how are these things controlled? There has to be some kind of feedback loop involved. And so more or less it's going to follow this simple reflex pathway where we start with a stimulus, a sensor picks it up, sends that information to the integrating center, and then a message sent to a target to create a response. And then, most importantly, there is a feedback loop that affects the stimulus. And so an example of the nervous system is the idea that you have vision, you have eyes. Your eyes detect a disturbance, that your eyes will then relay that information to some integrating center in your brain, and then your brain will then output some electrical signal for you to respond to that disturbance. So, for instance, you see a baseball flying towards you, your eyes collect that information, those are the sensors. Your brain says, you need to duck, and then your brain sends the signals to your body to activate those muscles for you to duck. So that's an example of a signal pathway that's being, controlled. And then, eventually your vision sees that ball's not there anymore, you can resume what you're doing. Endocrine system works somewhat similar. Cells will detect levels of chemicals. So for instance, here in the pancreas, these alpha and beta cells will detect levels of glucose in your bloodstream. And if it's too low, they're going to put out glucagon. If it's too high, they're going to put out insulin. And so this is also them detecting levels of a certain chemical and then putting out a signal based off it and then deciding to stop the signal whether levels plateau or not.